Hello, good evening to all of you. This is Anand Babu from Vijayawada, Andhra Pradesh. I am the EAMCET educator. Today, with another topic, I come before you. We are revising the topic that is cell, the unit of life. So, from this chapter, we are discussing about the questions based on NCERT, discussion on NCERT questions. How these NCERT questions are important. Already you are experienced with that in NEET examination. So conceptual understanding is very important. Okay. So each and every bit you see here carefully. That is very important for you. Related to that, number of bits you can get. I mean, the clarity about that bit. So one point you have to remember while discussion is going on. Related to that question, how you have to get the perfection. So watch carefully and listen and understand the concept, so that will make you to perfect in examination. Okay, before starting our program here, already I introduced myself, Anand Babu. My referral code on Unacademy Babu Live. On my referral code here, you will get 10% discount. And before starting, again it is a formality to explain about our plus subscription. And this is one of the Indian's best educational organization here, very big organization running with experienced educators. So continuous interaction helps you to make you perfect in all aspects as far as subject concerned. And this is a platform where you will learn about each and every topic, okay, doubt clarification, and also test, a number of tests at at regular intervals. Okay, this is convenient plan for you, affordable only. So I advise you to take plus subscription and also recommend to your friends to take this Unacademy subscription. Okay, thank you very much. Today our program is on cell, the structural and functional unit. Let us start with our program here. This is the first question you are facing here. You are facing here, first question. The most likely method used to determine the structural details of a cell organelle. Structural details of the cell organelle here. One you have to understand. Okay. So here 4 options are given here: autoradiography, microdissection, electron microscopy, phase contrast microscopy. Here what is the perfect answer here? And correct option here, the electron microscope. Electron microscope will magnify the object very smaller. Am I correct or not? Very smaller here. And it magnifies the object from 4,50,000 to 5,00,000 or 3,50,000 to 4 lakhs. So it magnifies the object by 3,50,000 to 4 lakhs. And its resolution power is 1000 resolution power. 1000 resolution power. Okay. So electron microscope, with the help of electron microscope, we are able to see tiny objects, Cell is very, very small, though, so still smaller parts are there inside the cell. So this is possible to see those structure with the help of this electron microscope only. Okay, and another question here. What are soluble pigments? What are soluble pigments here? What are soluble pigments found in plant cell vacuoles? What are soluble pigments? The right answer here is anthocyanins. Anthocyanin. Anthocyanin here. And where are anthocyanins are present here? This is vacuole here. Vacuole concentration is generally more than cytoplasm. Concentration is more than cytoplasm. Here pigments like anthocyanin and proteins, amino acids and waste materials are dependent. So this is nothing but a garbage of the cell here. It will accumulate all the materials and concentration of cell increases. Okay, because of this vacuole only. And also vacuole is very, very important to allow the water from outside. Low concentration to high concentration water. That part you have to understand here. Here, along with this materials, anthocyanin is one of the pigment present. This anthocyanin is water soluble. Water soluble here. And it is responsible for red color and what is it? Red color, purple color, okay, and blue. So this anthocyanin is water soluble, giving different colorations, which is present inside the nucleus. And next, coming to another bit here. Which of the following is true for nucleolus here? Which is the true for nucleolus? Right answer here. Here is which is true about the nucleolus. Nucleolus is okay. Here this is the right option. It's a site for active ribosomal RNA synthesis here. Okay, ribosomal RNA synthesis. So RNA synthesis is very important for the formation of synthesis of proteins. Am I correct or not? So this RNA synthesis happens with the help of the cell organelle, that is nucleolus. Okay. So nucleolus is nothing but a ribosomal factory. Ribosomes are produced from this region only. That is a nucleolus here. Where it is present inside the nucleus in the nucleoplasm, a membranous cell organelle here. Here you see, takes part in spindle formation. No, microtubules here, important for spindle formation. And it's a membrane bound cell organelle. No membrane is present. Okay, this is also incorrect. Larger nuclei are present in dividing cells. It is also incorrect. Okay, more number of nuclei are present where cell is capable of synthesizing the proteins. So it is very important. Okay, ribosomal RNA synthesis. Next, coming to another bit here. SCR and RER can be distinguished here. SCR and RER. How SCR is different from RER? SCR is nothing but smooth endoplasmic reticulum. So here, right answer, I will tell you SCR. SCR is fat and protein synthesizing cells. Sorry. Here you see. Fat synthesizing cells here. D option, some technical problem here. That little bit inconvenience. Here you see this is fat protein synthesizing cells. Here you see this is a fat protein synthesizing cells here. So it is very important for where it is for smooth endoplasmic reticulum means containing no ribosomes. This is no ribosomes are present here. So rough endoplasmic reticulum contains the here is a ribosomes. That point you have to remember. This here is very, very important for protein synthesis. This SCR is important for lipid synthesis, and also some steroids are synthesized like testosterone, estrogen, like this. So this is fat and protein synthesizing cells, SCR. Okay. RER is meant for what? The protein synthesizing cells. Why? Because RNA, this RER contains ribosomes, granular, protein and lipid synthesizing cells are not this one. Okay. This fat and protein synthesizing cells, SCR is present. Next, another point here. It's found in the cell wall of due to lack of this. What is that? It's found in cell wall of due to lack of. Here you see this is secondary cell wall material. If you observe the cell here, primary cell wall, secondary cell wall. Here this is secondary cell wall. This is a primary cell wall. Primary cell wall. This is secondary cell wall. Okay, on the secondary cell wall, here and there some pits are present. Okay, these pits are nothing but here secondary cell wall is made up of some materials, suberin and lignin, like that. Here and there small, some regions are thin. Those thin regions are called pits. Those pits are very, very important. Okay, those pits here, this is a pit, this is another cell here. Here is a pits. Pits are very important to transfer some materials from one cell to another cell, and also some stimuli for cell signaling. This is very, very important. So these pits are nothing but present on secondary wall, where secondary wall deposition is thin. That is. So cell plate, middle is the outermost layer. Okay, it is made up of calcium, magnesium pictate and calcium pictate, and cell plate here. Afterwards, cell plate is formed centrifugally. Primary wall material, primary cell wall material here. So right option is D. Another important function. The number of mitochondria per cell depends on number of mitochondria present on cell difference. Number of mitochondria. That depends on physiological activity of the cell. Physiological functioning of the cell here. For example, cell function is more active in synthesizing proteins. Here number of mitochondria more. That point you have to understand. Number of mitochondria more here. For example, wings contain muscles. Wing muscles contain number of mitochondria, large number of mitochondria. Why? Because more amount of energy is required for flapping their wings. And also in liver cell, you see here, liver cell, heart cells, you see here, large number of these mitochondria are present. Okay, in a heart muscle cell here, nearly 40,000 mitochondria are present. You know very well, mitochondria are the sources of energy. So, more amount of energy present in that heart and liver. Why? Because their functioning is very important in our body. Okay, activity of cell is more. Another bit here. Select the incorrect statement here regarding the plasma membrane. Here the right option here, 52% here. See here. Here 52%. 52% here is 52% proteins and 40% lipids constitutes the membrane of human RBC. So this is not here. Regarding the plasma, it is incorrect here. 40% here proteins and 52% lipids. That point you have to understand. Okay. A ratio of proteins and lipids varies continuously in different cell types. Yes. So this is incorrect. Arrangement of proteins and lipids LPPL. I mean, this is protein layer. This is another protein layer. In between, two lipid layers are present. This is also correct. Head of lipid is hydrophilic. So here is a lipid part as two parts here. This is head and this is a tail. It is made up of cholesterol, no, unsaturated fatty acids. So lipid head is hydrophilic. It is water loving. This portion is hydrophobic. Okay, this portion is hydrophobic. And next, coming to another bit here. Integral cell membrane proteins. Integral cell membrane proteins, partially embedded in lipid layers or completely embedded, show lateral but not vertical movements within bilayer lipid. All this. What is the right option here? Right option here, all of these only. Here you see partially embedded in the lipid layer. You see here like this. Integral proteins. Integral proteins are carrier proteins. Okay, or partially embedded in a lipid layer like this. Completely or partially. And also here proteins move laterally, not vertically. Lateral movement of this protein is very important. Okay, for normal functioning of the cell membrane. Lateral movement of protein happens here. So fluid mosaic model of the cell membrane we have to study. So all of this is answer. Next, coming to which organelle is not a part of the endomembrane system here. Which is not part here. So here this is mitochondria is not the part. Okay. Endoplasmic reticulum, Golgi complex, lysosomes are functionally coordinated. For example, this is ER and this is what is called Golgi complex. Okay, and these are lysosomes here. Okay, the three cell organelles are interrelated here, coordinated. Here proteins are synthesized by this endoplasmic reticulum, ER, and these proteins are modified in where? Golgi complex, and modified proteins are packed and are transferred to targeted cells here. One of the vacuole is larger, containing these proteins here, modified protein. This is what is called lysosome. So functionally all are related. Those cell organelles which are functionally coordinated, they come under endomembrane system. They come under endomembrane system. So mitochondria is not involved. Independently, the mitochondria will work. So its important function is oxidizing the food material, releasing the energy in the form of ATP. So this is incorrect here. It is not coming under endomembrane system. Another bit here. Which of the following is correct for the origin of lysosome? How these lysosomes are originated here? These lysosomes origin. Here you see first endoplasmic reticulum. Already I told you. Then Golgi body, Golgi, then lysosomes. From the Golgi complex only, these lysosomes are developed. So this is a Golgi complex. Modified proteins here are packed, and in the form of lysosomes, they are released. Okay. So Golgi body. These other options B, C, D, all are wrong here. Okay. Next, coming to another bit here. Which of the following represents the features of lysosomes? Which of the following is related to lysosomes here? Here you see one more important point. So this is the right answer. A lower pH than the cytoplasm. Here what is the important feature here? The lysosome is present inside, and this lysosome is acidic. It will active under acidic medium only. So this is pH 7 here. Below 4.5 pH it works. It is active under 4.5 value pH here. Acidic in nature. So this is the lower pH than cytoplasm. Its pH is lower than the cytoplasm here. This is cytoplasm. Okay. So that is related to these lysosomes. A lower pH than the cytoplasm. Okay, reduced hydrolysis activity is wrong. Okay, why? Because enzymes are present over here. Hydrolytic enzymes are present. They are very active and important for intracellular digestion here. Okay, double membrane envelope, no, single membrane is present. So this is the right option. Okay, another important bit. Which are the following statements regarding sphaerosomes? Here, see sphaerosomes. Yes, sphaerosomes. Important here, sphaerosomes. Here, see, these are sphaerosomes. Is not correct here. Regarding sphaerosomes, not correct. Sphaerosomes here, the D is the right option here. D is the right option. Here, this is the D is the right option. Take part in the photorespiration. This is incorrect here, not correct here. Photorespiration, which is the cell organelles, microbodies involved means here peroxisomes. Peroxisomes, mitochondria, and chloroplast. These three cell organelles involved in the photorespiration. Okay, but sphaerosomes have no relation with this photorespiration. Okay, take part in synthesis and storage of lipids. Correct. Important for storage of lipids and this synthesis. And also this is bounded by single membrane. The sphaerosomes covered by single membrane. Abundant in endosperm cells of the seeds. Okay, so these are all correct options here, excepting this fourth. Next, coming to another bit here. Which one of these is not correct regarding peroxisomes? Peroxisomes. Sphaerosomes over, now peroxisomes here. Which one of these is not correct regarding this? Here this is C is not correct. Take part in the synthesis and storage of lipids. And synthesis and storage of lipids is the important function of what? Sphaerosomes. Just now I told you, sphaerosomes. So it is also a single membrane, bounded cell organelle, and photorespiration here. The cell organelle involved in photorespiration, peroxisomes, protect the cell from the toxic effect of the H2O2. This is also an important bit here. In chemical reactions here, electrons are liberated. No, those electrons are transferred to oxygen and forming H2O2 here. H2O, hydrogen peroxide. Okay, accumulation is harmful in the cell here. Harmful in the cell. So H2O2, hydrogen peroxide, should be paralyzed. Broken down. This happens with the help of these peroxisomes only. How in peroxisomes one enzyme is there, that is catalase enzyme. Those catalase enzyme here break the hydrogen peroxide into what is it? Hydrogen and this hydrogen and oxygen. Okay, like this. So that accumulation of hydrogen peroxide will not be there here. So protect the cell from the toxic effects of the H2O2, hydrogen peroxide. It's here capital H. Okay, another bit here. Unicellular microscopic organisms were first studied by whom? Right answer here is Leeuwenhoek. Many people have confusion here. Robert Hooke, they put the answer Robert Hooke. What is about Robert Hooke? Robert Hooke discovered the cell. Okay, this is one point. Another point, this Robert Hooke observed dry cell only. That is another point. Okay. He observed compartment-like sections. He took what is that cork section like this. So these are related to Robert Hooke. Priestley, you see here oxygen discovered by him. Pasteur, Louis Pasteur, you know very well, pasteurization like this. Leeuwenhoek, he observed microorganisms like bacteria, okay, some amoeba-like organisms, okay, in pond water like this. So he was the first man who observed unicellular microscopic organism. And also about Leeuwenhoek, living cell identified by Leeuwenhoek. Okay, Robert Hooke identified the dry cell only. That point you have to understand. Next, coming to cell theory was formulated by. This is a direct question here. Easily you can attempt the answer. Schleiden and Schwann. According to Schleiden and Schwann, plant tissues, animal tissues are made up of what? Living cells. This is a cell theory. Okay. And here about Schwann here, one more important point. He observed cell membrane and cell wall also. Cell wall and cell membrane also observed by him. Okay, Schwann. Yes. And Robert Hooke, you know very well. Robert Hooke. What is invention Robert Hooke here? Robert Hooke. Sorry. Robert Hooke here. The cursor is not moving here. Robert Hooke here. So Robert Hooke here. I will take another pen. Here Robert Hooke, you know, discovered the cell. He discovered dry cell, Leeuwenhoek living cell, Marcello Malpighi, initially plant tissues. Okay, plant tissues. And another bit here. Who proposed a modification in the cell theory? Who proposed modification? Who proposed here modification? Who proposed modification here? First, Schleiden and Schwann, Rudolf Virchow, and Robert Hooke here, Marcello Malpighi. Rudolf Virchow is the right answer here. Rudolf Virchow. According to Rudolf Virchow, new cells born from pre-existing cells. Rudolf Virchow. According to Rudolf Virchow here, you see. So this is pre-existing cells from which new cells born. This is cell lineage theory also, it is called cell lineage theory. And also omnicellular unicellular. Omnicellular unicellular. So Rudolf Virchow is the correct answer. Schleiden Schwann, cell theory. Robert Hooke discovered the cell. Marcello Malpighi, you know very well, plant tissues. And dash are self-replicating extra chromosomal segments of double-stranded circular and naked DNA present in the bacterial cell. So here plasmids. In some of the bacteria you see, in addition with the DNA, this is DNA, addition with DNA, small circular DNA will be present. This is what is called plasmid. So this plasmid is very, very important. It is used in recombinant DNA technology. So here now useful genes will be present, antibiotic resistant genes and NIF genes like that, nitrogen fixing genes. Okay, those genes will be extracted, isolated by using the technique that is rDNA technology. Insulin producing genes also present. Sure, so plasmid is very, very important. And nucleoid, you know very well where nucleus is present. Mesosomes are what? And here in bacterial cell membrane having here and there like this infoldings. These infoldings are called mesosomes. Okay, these mesosomes believed to be involved in respiration here. These are involved in respiration. Why? Because these mesosomes contain enzymes. Those enzymes are useful for respiration here. So, mitochondria are not present in the prokaryotic cells now. Mitochondria are not present here. Okay. So, as mitochondria are not present, okay, instead of mitochondria, mesosomes are present. Bacteriophages means virus attacking the bacteria like this. And another important point, What is the true about genetic material of a prokaryotic cell? Lacks histone proteins, not enveloped by nuclear membrane, composed of a single circular and naked DNA molecule, all of this. So here easily you can tell the answer here, D is right answer. Lacks histone proteins. Histone proteins are what? Histone proteins are present in the eukaryotic DNA only. Histone proteins mainly lysine, amino acid, and arginine. It consists of this. It is made up of mainly lysine, arginine, amino acids. These histone proteins are present only in eukaryotic DNA. Eukaryotic DNA. In prokaryotic DNA here, histone proteins are lacking. That point you have to answer. That's why, okay, naked DNA. It is naked DNA. Why it is called naked DNA? Histone proteins are absent. In eukaryotic DNA, histone proteins are present. Important for what? Packaging of DNA. Packaging of what is it? Packaging. These histone proteins are important for packaging of DNA here. Okay, why? Because histone proteins are having plus charge. This negative charge. DNA is a negative charge. So composed of a single circular and naked DNA. Naked DNA. Why it is called naked DNA? Means here histone proteins are absent. Okay. And coming to another bit here. The figures of cork cells as seen by Robert Hooke were published in the book. The name of the book here is Micrographia. So this is a direct question here. Origin of species, origin of species Darwin, and species Plantarum, Linnaeus, genera Plantarum also Linnaeus. Okay, Micrographia book was written by Robert Hooke. Another bit here. If you remove the fimbriae from the bacterial cell, which of the following would you expect to happen? What is the right answer here? Here you see, the bacteria would not adhere to the host tissue. Here fimbriae are the tube-like structures here. With the help of that, it will attach to here. What is that? Host cell or tissue here. These are fimbriae here. Without fimbriae, it is unable to attach or adhere to the host tissue. Okay, that is the right answer. Transportation of molecules across the membrane. So these are all not related here. Another question here. The type of ribosomes found in prokaryotes. Easily can tell, direct answer only. Answer is here 70S type. Okay, 80S type and 70S type both are present in eukaryotic cell. Am I correct or not? 80S. Where 80S present in eukaryotic cell? 80S present in the cytoplasm. Okay, and also rough ER or ER. 80S only. 80S. But whereas in mitochondria, you see here, mitochondria and chloroplast here. In eukaryotic cell, chloroplast. Some technical problem here, cursor is not functioning. Mitochondria and what is it? Chloroplast here. Chloroplast here, 70S ribosomes are present. So you have to understand what is that. In eukaryotic cell, 80S, 70S ribosomes are present. Whereas in prokaryotic cell, only 70S ribosomes are present. 80S ribosomes are present in eukaryotic cell in the cytoplasm and also on rough ER. And in mitochondria and chloroplast, 70S ribosomes are present. Okay. So this is the clarification for you. According to unit membrane structure, the thickness of plasmamembrane is about. What is the thickness here? This is the plasmamembrane here. Two are lipid layers. This is protein layer. These two are bilipid layer. This is protein layer. I mean, bilipid layer is sandwiched between the two protein layers. Here, this is protein layer. This is protein layer. So here, what is thickness? 75 angstrom. 75 angstrom. Here, this is what is that? 35 angstrom bilipid layer thickness, and protein layer 20, and another protein layer also 20 angstrom. Totally, here is 75 angstrom thickness it is having. Okay. Another important bit. A phospholipid molecule is amphithetic and produces two layers coming in contact with water. The head of phospholipid molecule is here. So it is amphithetic. Amphithetic, what is the meaning of the amphithetic here? So for example, this is the lipid structure here, consisting head and tail here like this. The head part is hydrophilic. Hydrophilic. Okay, and this is what is called hydrophobic. This is hydrophobic. Okay, so the head part is towards the outer side, and this hydrophobic part of the tail is towards the inner side like this. Okay, that part you have to understand. So mostly in cell membrane, lipid is only the present, phospholipids. Okay, that point you have to understand. So here, this is correct answer. In the inner side, embedded in the protein molecule at an outer surface here. That part is towards the outer surface. And another bit here. The best material for study of structure of cell membrane is, what is that answer here? RBC of human beings. Why? Because in RBC here, nucleus is not present, other cell organelles absent. So this type of cell is very convenient to study about the structure of the cell membrane. So here the cell membrane will be studied very clearly here. RBC also covered by one membrane. So other options are incorrect here. Which chemical property is shared by all types of lipids forming the plasma membrane? So this is hydrophobic region here. Already I explained this one. Which is the best way to separate intact chloroplast? Separation of chloroplast from the green leaves with the application of what is the solution here? Alcohol. Alcohol is the best solution here to isolate the chloroplast from the green leaves of the angiosperms plants here. And the lipids are arranged within the membrane with, already I told you, polar heads towards what is that? Both heads and tails. Heads towards the outer sides and tail towards the inside. Already explained this. Both heads and tails towards inside. This is wrong, and also this is wrong. This is wrong. Okay. So another important. The most abundant lipid in the cell membrane is, here cell membrane most abundant here, phosphoglycerides. Phosphoglycerides. Okay. Cutin, glycolipid, steroid. These are not related to this. So direct answer this is. According to the modern concept, cell membrane is quasi-fluid here. What is it? Quasi-fluid. Here this is the quasi-fluid. Semi-fluid nature here. Semi-fluid nature is very, very important here. For the lateral movement of the proteins, proteins move laterally here. Lateral movement of the proteins gives semi-fluid nature. This is semi-fluid. Because of semi-fluid nature of the cell membrane, so all the properties associated with the cell membrane will take place systematically. Okay, what are the properties associated with cell membrane? Okay, another important bit. Cell membrane is selective permeable. This means that is easily you can say this is the answer here. Allows only some substances. It will not allow all substances to pass through. Only some of the substances will be allowed by it. So that is the nature of the cell membrane here. Okay. Thank you very much. Time is up. Thank you for your patience.